Hi, my name is Zach, and I'm with Team BCM. Our project this semester has been to discover new genomic signatures across stages of a common vision-related disease. But first, let me introduce you to Rosa, an elderly woman who has recently had trouble seeing her grandchildren clearly and no longer gets enjoyment out of her usual nature walks. Rosa needs our help because she has just been diagnosed with AMD, or age-related macular degeneration. AMD is caused by a deterioration of cells in the retina, a region of the eye whose function is to convert light into neural signals that are capable of producing a clear image in the brain. The disease can be symptomatically broken up into 4 stages, where the first stage indicates no symptoms, and at its most extreme stage, AMD causes central vision loss, which explains why Rosa cannot see clearly. We want to help because there are currently few viable medical treatments for AMD, and because the disease is hard to detect, even in its late stages. In the figure below, we have sampled the same 25 genes each from a patient in stage 1 and stage 4. As you can see, there is hardly any difference in the expression level of each gene across the 2 patients. Finally, AMD is a major cause of vision loss in the elderly. About 1 in 4 people over the age of 65 suffers from AMD, affecting 11 million people in the US alone. Our project seeks to detect AMD in ways that nobody has done before, so that we can identify the affected genes and pathways, enabling a fast track to new treatments and cures. We were provided data for 453 patients, each assigned to one of the 4 stages of AMD. For each subject, we were given their gene expression counts for about 58,000 genes. Our initial data is too high dimensional to perform an analysis on, so we resorted to low variance filtering and further ANOVA analysis to reduce our space to about 4,000 genes per patient. First, we remove redundant genes based on their low variance across stages, and with ANOVA, then we examine the differences between each group to determine which genes stand out the most. We also perform targeted dimensional reduction by inspecting the effects of known batches, such as age and sex. For example, when we remove genes on the X and Y chromosomes, we no longer see a clustering division when labeling by male and female. As an effect, we are left with a dataset with its batch effects and artifacts removed, but our stages of AMD are still inseparable by mere clustering. I'll hand it off to Minjun.
Our first objective is to extract a set of genes that are most important for each stage of AMD. At first, we try different feature selection methods to find the best model for multi-classification problem. We try multinomial regression with Lasso. In this model, the tuning parameter lambda is chosen by cross-validation, and the model chooses the lambda where multinomial deviance is minimized. We also use tree-based algorithms such as random forest and XGBoost. For stability testing for selected genes, we did bootstrapping with 500 iterations. We also cross-checked important genes that are chosen by all 3 models, and our models identify genes MOXD1 and PMET1, which genes are verified to be differentially expressed genes. To validate our model, we reported tenfold cross-validation accuracy for predicting 4 different AMD stages. Among 3 models, multinomial lasso regression had the best performance of 60.2%. The model missed stage 4 the most, and we thought this is interesting because stage 4 is the most advanced disease. We also used only 2 stages, stage 1 and stage 4, as they're expected to show the most different gene expression. Our model predicts stage 1 with high accuracy, but predicts stage 4 with low accuracy. Now, we touch holdout data of 46 patients, and the model predicted with 34.8% accuracy for all stages and 47% for predicting stage 1 and stage 4. In both cases, the model could not predict stage 4. One reason is that we have small samples for stage 4, but we analyzed further to understand why the model cannot predict stage 4 with high accuracy. We first looked at cluster heatmap of 20 most important genes in stage 4. Each row represents the gene, each column represents patient. In the red box, gene expression is mostly black, so these genes have low expression and have low power to predict stage 4. We also looked at correlation matrix of 40 important genes in stage 4 to see if genes are correlated to each other. Most genes have correlation coefficient above 0.8, and this is another challenge that the model could not choose genes that are independent to each other. With these challenges, we explore genomic pathways and their association with AMD stages, as well as pathway networks. A pathway is linked interactions among genes that lead to a change, and genomic pathway is a functional pathway that describes how genes work together to activate or repress genes in a cell. Once we identify the important genes through our model, we can now look at the complement pathway to see if any genes are disrupted by affected genes. With Go Enrichment Analysis, we obtained top 10 pathway for AMD stages. Here we found pathway called positive regulation of immune complex clearance by monocyte and macrophages, and this pathway corroborates with role of immune response in AMD biology. For our next step, we want to introduce multiple pathways such as transcriptomics, epigenomics, or proteomics, and this network analysis will give us holistic view of the pathways. Thank you very much.